we know we've got a lot of people online that are excited to hear this and to get our industry panel going. So I'm going to introduce to you yet another amazing individual who is with the Angelman Syndrome Foundation. She is our Chief Medical Officer, who is leading all of our clinics here in the United States and supporting the ones we have globally. If you don't know about the Angelman Syndrome clinics, we will be talking a lot more about that tomorrow when we have our clinical panels, when we have our seizure panels and other panels. So tomorrow is all about the clinical part of Angelman syndrome, but we would love to help you get access to a clinic if that's important to you and that's a need for you and your family. But Dr. Elizabeth Gelazo is our Chief Medical Officer, and she's going to help bring our industry up to start giving industry updates, and then we'll have a panel afterwards. But Liz, if that's okay, has been a part of the Angelman Syndrome Foundation as a board member, but more recently moved over in this role to help us really expand what we're doing with the Ladder Learning Network. And thank you, Allison, for putting a plug in for the Ladder database. If you want to learn more about that, there's a table out there. So it's definitely important for families to get engaged with that. But she's also a mama, like all of us out here, and this work is meaningful and passionate for her as well. So I'm going to invite her to the stage, and we're going to get our industry updates going. So let's give it up for Dr. Elizabeth Gelazo.
Thank you, Amanda, and thank you to all the families, both here in the room and joining us virtually. It is my distinct honor to get to introduce our industry colleagues and kick off our panel where we'll hear updates from not all, but many of the pharmaceutical companies who are active in our space right now, bringing these clinical trials that I know have generated so much hope in our community. So without further ado, I'm going to look over here to my right to our colleagues at Roche. I'm inviting Ina Trabert to the stage to talk to us about updates from the Roche development program on rugonersen for Angelman syndrome. Come on up.
So thank you so much, Liz, for the introduction. And I'm really very, very grateful and honored to be here to kick off the session, really. Also to be here with you in the room. Like I'm really speaking on behalf of Roche today. And I can just tell you that we are very energized to be here with you in the same room with so many families, see your children. It really gives us a boost of energy. So yeah, my name is Ina Trabert, and I'm leading the early development team at Roche for our compound for Angelman syndrome, for rugonersen. It's really a group of very passionate people working hard on this, sometimes day and night. I also would like to send you greetings from my colleague, Brenda Vincenzi, who is the medical director on this program. Some of you might have seen her, and she will be in touch with the community going forward. So she will be on the scene for you as well. So we really very much appreciate that many of you might have been looking forward to now hear from Roche, and especially in light of the recent announcement of Ultragenyx, you must have been waiting to hear an update. So I'm very, very grateful to talk about this today and to have this opportunity. So I would like to start by telling you a little bit about our journey that we've been on with regard to basically the clinical part of our research in Angelman syndrome. So we've really started in 2015 to engage with the community, to start to learn about the burden and to really learn what it really means to live with Angelman syndrome and what really matters to you people. We've then moved on to work on the concept model that we've heard about. And in 2018, we have been starting our clinical studies on Angelman syndrome. We started with an observational study that many of you have heard about, maybe were on, called Freesias, which I will be talking about a little bit later. And then in 2020, we've started our actual drug program that I will be giving you an update on. So our phase 1 study called Tangelo, that's a study in children 1 to 12 years. And this study has actually 2 parts. And the news that I can bring today to you is that this year we've been able to complete the first part of that study, which is the dose finding, dose escalation part. And we've also been able to start the second part of the study, which now looks into chronic dosing. So we've now been starting to actually dose patients chronically, long-term with our drug, which is important. And I'm happy to be able to report on this today. Also, we've been running a so-called PET study, which is an imaging study that's actually able to visualize the drug upon lumbar puncture as it travels to the brain. And we have been able to derive important information from the study as well. So overall, I think with this, it might be fair to say that this might be one of the largest, a large program for an ASO and Angelman syndrome right now in industry. The commitment at Roche is really huge. We understand the urgency, we really do, and we are committed to speed. We absolutely try to bring this drug to you patients, to your families, as fast as possible. We're absolutely committed to go unconventional ways. The one thing that we won't sacrifice for speed is science, a strong science, robust science, because we absolutely believe that this is key for success. So let me just talk a little bit about our observational study. But actually yesterday at the research conference, there was a more comprehensive presentation on this. It's been completed now, so it will be published soon. So the data of the study will be available to the community really soon. This study has been run in collaboration with Biogen and Ionis. It was a pre-competitive collaboration. I just wanted to use the opportunity to point out to all of you that actually GeneTx is the word under which, the term under which Roche operates in the US. So just to be clear, it's just a similarity of names, but not to be confused with GeneTx or GeneTx, which was just acquired by Ultragenyx. GeneTx and Roche is one and the same company, just to make sure. The Frisia study had really the objective to explore and investigate assessment tools that can be used reliably and sensitively to measure symptoms of AS and then ultimately to measure a treatment effect, a treatment response, as we've just heard from the previous speakers. The objective of the study was really met. We have a very valuable data set from Frisia's that we can use. We've identified some tools that we will be taking forward into drug development, and there are other tools that we've discarded, which was actually the plan. So I won't go into any more detail, but I really would like to use this opportunity to say, despite COVID, the study was quite a lot impacted by COVID. And despite the fact that patients actually don't directly benefit from such a study, we were able to complete it pretty much according to plan, thanks to you all, thanks to your commitment, thanks to everybody involved. So just a big, big thank you to everyone who supported this endeavor. So with this, I'd really like to talk to you about our drug program. So the first piece of news is that our drug now has a name, rugonersen, which is kind of nice because it's a bit like a baby that suddenly has a name, so it's a bit easier to relate to it. We kind of enjoy talking about it. And also in the life cycle of a molecule, it really marks or it indicates that progress has been made. So it's kind of nice. So rugonersen is an RNA therapy, an ASO. It's also an LNA. So if you hear those terms, they can be used interchangeably. Both are correct. That is able to, has the potential to unsilence the paternal copy of UBE3A. You've heard about this before, and therefore is able to express UBE3A protein in the brain. Now, there was another presentation yesterday by my colleague, Thomas Kramer, who yesterday at the research conference showed unambiguously that this particular molecule is able to really induce long-term expression of UBE3A protein in a monkey's brain upon a single dose. That's quite remarkable. We do consider this as a prerequisite to actually take such a molecule into development. So quite an important milestone for us. So just to really emphasize this point, despite our phase 1 study, Tangelo, being run in children 1 to 12, There is an aspiration at Roche to bring this drug to everybody with Angelman syndrome. We really have this goal. It cannot be a promise at this point because it will be a data-driven decision, but there absolutely is this aspiration to bring this drug to everybody as fast as possible. We are already exploring infrequent interventions in the long-term extension study, as I will be showing you in a minute. So our TANGELO phase 1 study is in participants 1 to 12. We are able to recruit up to 74 patients with Angelman syndrome mutation and deletion genotypes. The study is running in the US and in 3 countries across the EU. It has started in 2020, and now that the long-term extension part of that study has started, it will continue to run for quite some time. The end of the study is actually flexible, might be data-driven, which is why it says approximately on the slide. So just to explain to you a bit more what the TANGELO study really does. It has these 2 parts I have mentioned before. Part 1 was really there to identify safe and well-tolerated doses that we can take forward into long-term dosing. We have recruited 51 subjects to date. We have quite a large data set, and we have identified and selected doses that we are now able to take forward into the chronic part, into the long-term extension. In the long-term extension part, patients will receive a dose every 16 or every 24 weeks. So we are really happy to basically be able to report to you that we are exploring these very patient-friendly intervals of doses and are looking at the effect that the drug has on patients when they take it long-term. So that's what I can share about the study. There's one thing that I cannot share, which is data. So at Roche, we really care about data validity, and there's a very specific reason why we are not sharing data of this ongoing trial prematurely. We've been hearing about placebo response before, so I really want to, instead of sharing data with you today, I really want to take a moment to explain to you, and hopefully you can understand and buy into these reasons why we are not sharing data, although we all share the same wish that all these drugs really work, right? So we share the same curiosity, but we need you to bear with us for a little bit longer. So let me just explain to you, in order to explain why we're not sharing data, what a placebo effect is. And, you know, I can build on what my previous speakers have already talked about. So just in general, positive beliefs as well as negative beliefs do influence trial results all the time. That's what's happening on clinical trials. Now, some of those trials, especially phase 3 trials, registrational trials, are placebo controlled, which means you have a group of patients on placebo and you have a group of patients on drug. All of those people will improve. They will have measurable effects, clearly. And in the group of people who take the drug, you will have a combination of a placebo effect, of an expectation effect, expectation bias, and a drug effect. Now, you might imagine that the bigger the placebo effect, the more difficult it is to detect a drug effect. And this is really something that we are concerned about. We really need everybody to work together to keep the expectation bias low. So, which is why we're not sharing data at this point, because it would be premature. The study is ongoing. We don't want to further inflate expectations. So there's really an ask that I want to close my 10 minutes presentation with to all of you, which is we all need you to please stay as objective as possible about potential or suspected drug effects at this point. We also ask you to not jump to conclusions only because Roche is not sharing data, right? So us not sharing data is not a good sign. It's not a bad sign. It doesn't allow for any conclusions about the data itself at this point. Also, we are committed to transparency. Definitely, we are willing and happy and committed to share data with you if, given what I just explained, the time is right and appropriate, then we absolutely will be sharing data with you. So with this, I would like to really ask you to follow this. And I know we're going to talk about this a bit later in the panel as well. So I really would like to end by thanking you. Thank you for your trust, for your confidence in our program, and thank you for your attention right now. Thank you.
You, Ina, and the Roche team. Next up, I'd like to invite Camille Bedrosian from Ultragenyx to provide us an update on the current status and clinical results of the phase 1/2 study of GeneTx 102 in individuals with Angelman syndrome.
Thank you so much, Amanda. It's a great, great pleasure to be here. I'm sorry, Elizabeth, and Amanda also for being here today to share with you some initial preliminary clinical results from our phase 1/2 study of GeneTx 102. I want to thank you for this opportunity and also thank the participants, the families, the investigators and their teams and advocacy really for putting their trust in this trial and going forward as well. It's sort of a thank you in advance also. So GeneTx 102, I'll speak to that in a moment. As you heard from Allison just a bit ago, recently Ultragenyx did acquire GeneTx, which is the company that's been developing GeneTx 102, and we've partnered with them since 2019 in the development of this antisense oligo. Our acquisition of GeneTx is a reaffirmation of our deep commitment to the Angelman community to find a solution for this disease, and it may be more than one solution, to be clear, as you heard earlier. We are all in for Angelman syndrome, and now our deep focus is on developing GeneTx 102 further and taking it onward. So you'll see here the schematic for GeneTx 102, which is still investigational, and it's in early stage phase 1/2 studies. Cohorts 4 and 5 here represent 2 cohorts of patients by age: younger children, 4 to less than 8, and older children, cohort 5, 8 to less than 18, studies ongoing in Canada and the UK. And you can see the schema for the dosing, monthly dosing as loading, intrathecal administration, followed by then every 3-month maintenance dosing. Important to say that we wanted very much to be, to have this update to be clear on the fact that we have identified an approach to dosing GeneTx 102, both from the starting dose and the gradual dose escalation that we are doing now, as you'll see in a moment, as well as by modifying the way we administer the drug, by having patients tilt with their head down to help with the mixing of the antisense up to the brain, and also a flush to further dilute and mix the antisense. So 14 patients have been enrolled so far with this new approach, starting at very low doses, 3.3 in younger patients, 5 milligrams in older patients. And you can see based on the cohorts here, how far patients have come as of mid-July. Importantly, half the patients have a cumulative dose of over 20 milligrams, a single dose of which did cause lower extremity weakness before. And virtually all of the patients have gone a longer interval of time from their first dose than those patients of the original 5 who experienced lower extremity weakness. You'll see also we have a study ongoing in the US led by Dr. Barry Kravitz, in which 2 milligrams flat dose is administered monthly loading and then every 3-month maintenance dosing. And this dosing continues as we speak. You'll see also that we're now up to 7.5 and 10 milligrams in these patients. And that's important to keep in mind as we go forward. First and foremost, we are seeing a very tolerable tolerability with the dosing regimen that we have now and the doses. No drug-related serious adverse events, no adverse events or serious adverse events of lower extremity weakness. And there have been some unrelated SAEs, a varicella-zoster reactivation, that's chickenpox reactivating, nausea and vomiting after swallowing an ECG lead, curiosity, and that's something that we acknowledge and expect, and then some vomiting. And in the era of the 2020s, COVID is a relevant adverse event, although certainly not drug related. I'll share with you a bit of data here, again, very early, but promising data. CGIC, that's the Clinical Global Impression of Change for Angelman Syndrome. We used to refer to it as improvement, but that was presupposing that everyone would improve, possibly. There is the possibility, as you can see in the lower bars there, that patients could worsen. So it's really change, and change relative to baseline. And you can see the 5 domains that we're studying: sleep, behavior, communication, gross and fine motor, and then an overall impression. Importantly, the top row of graphs represent the cohort 4, 7 to less than 8, and the lower bar, cohort 5, the first 3 patients there for the 8 to less than 18. And you can see that 7 of the 9 patients of these combined cohorts have shown improvement from baseline in at least 3 domains. And that's as of after the 6 weeks after the 4th loading dose. Now I'm going to dive a bit more deeply and show you bits of data from the various domains, utilizing different tools to assess outcomes. And you heard a bit about outcomes before, and a lot of work has gone into determining what measures and tools to use, and also who is doing those assessments. Because for some of the assessments, like the CGIC, the clinician, PI does the assessment. Here we're going to look at severity. Again, the clinician does the assessment. And this is severity. It's somewhat more objective than the relative change because it's assessed at that moment based on very specific criteria, 2 of which you see here. And then after scoring, the physician will go back and see how much the severity has improved. Improvement in this case is minus numbers because the severity is decreasing. The bars are pointing up as a symbol of improvement. And you can see for sleep, quite a few of the patients have exhibited improvement in sleep, which is, and you can see from the commentary from caregivers, very important, going from sleeping 3 hours a night to over 12 hours for one of the patients, the patient in blue. We continue to follow that domain, of course, using other measures as well. Then we use the Vineland-3 for behavior. And here the clinician interviews the caregiver to have their assessments of various domains. And here it's the self-care domain, the personal domain of behavior. And you can see across both cohorts, 4 on the left, 5 on the right, that there numerically is some improvement in most patients, and 2 of the patients actually showed statistically significant improvement. And again, this is 6 weeks after the last loading dose, and you can see some of the commentary there. The next tool used, and we're going to look at communication, both receptive, which is, does the individual understand, and expressive, can the individual communicate their what they're feeling or what they need. And the Bailey is a very objective score conducted by therapists who have specific tasks that they ask the participant to conduct. Here you can see the receptive communication, younger and somewhat older children, and then expressive on the right-hand side. You can see some already improvements for most patients, modest to be sure, with 2 statistically significant at day 128. And then 6 weeks after that, before the next and the first maintenance dose, you can see further improvements in these scores to statistical significance for a subset of the patients. This illustrates for us not only the importance of dose to knock down that antisense and allow for UBE3A expression, and also the importance of time to allow for the knockdown and the expression of the enzyme to work, do what it needs to do in the brain. Here is a heat map representation of those Bailey data. And you can see of the original 5, 3 of the 5 patients had statistically significant improvements in either receptive or expressive communication. And for the patients in Canada, UK, you can see that 7 of the 9 had, at the most recent assessment, statistically meaningful improvements in those receptive or expressive scores. And these changes really based on natural history data that all of you have contributed to allowing us to understand that really is not observed in patients. And I should mention these patients all have deletions. Another tool that we use is ORCA, and you can see similar movements, a couple patients with statistically significant improvement and then numerical improvement as well. And then fine and gross motor, some improvement there using the Bailey again with the specific tasks and an independent therapist to do the assessments. So in conclusion, and then we'll give a little bit about next steps, GTX-102, as we now are dosing the drug, has acceptable safety results at the current dose and administration without drug-related serious adverse events. We do see encouraging clinical activity, early signs for sure, with multiple functional domains showing positive movement in most patients. We've dosed up to 7.5 and 10 milligrams. And importantly, we have clearance from Canada and UK to start a new set of cohorts of patients, starting at somewhat higher loading doses, specifically 7.5 and 10 milligrams. So we're very excited about those possibilities as well. The trial is continuing in the UK, Canada, and in the US at 2 milligrams, and we'll be opening sites globally. We also are continuing to work with the FDA to allow for harmonization of the dosing strategy as well as administration. And finally, though not on the slide, I'm very gratified to share with you that the first of the original patients is now redosed up in Canada, thanks to the Canadian site up there, and that patient is doing well. And we will continue to have patients redosed as they wish going forward. And going forward as well, we will be planning for a pivotal study that's randomized and well-controlled, and look at additional patient populations as well. So thank you very much for your attention, and I look forward to the questions later.
Thank you, Camille, and the whole Ultragenyx team. I'd like to next invite up our team from PTC Therapeutics, led by Ed Weber and Afode Ajari, to give us an update on their gene therapy program in Angelman syndrome. Welcome.
Wow, it still gets me excited to stand in front of all you guys after all these years. I'm really excited, and thank you very much for the chance to come and talk about PTC Therapeutics and where we are on our gene therapy program. I just want to give a little bit of background. A lot of people don't know what PTC Therapeutics is about, and they're not as big as some of the other therapeutic companies that have come up here. PTC Therapeutics was established in 1998 by its current CEO, Stuart Peltz. We are a research-led organization, which really means that the research dictates where the targets are and where the clinical trials go. And it's really kind of unusual to be in a company with such a strong research background. We have a footprint in over 50 countries. We really are a global pharmaceutical company. PTC is dedicated to the discovery, the development, and the commercialization of numerous different therapeutics for one reason: to find good therapeutics for beneficial effects in rare disorders. I think that's pretty cool for a company. PTC has a pretty diverse pipeline. We do a lot of different research in different areas. We have a really strong commercialization, as you can see in the slide here. We do a lot of different types of research and everything from oncology. We do metabolic research, and we have a very strong gene therapy program. A lot of research has gone into this, and Angelman Syndrome is right smack in the middle of that, and a big focus for PTC. We say this a lot at PTC Therapeutics, that we're passionate about our purpose, and it really is true. We think of ourselves more than just a biotherapeutics company. This company has a long history, 20-year history of finding therapeutics and providing benefits for patients in the rare disease field. And I think it really kind of shows what direction we're going in and what we do and how we do it. I want to give a little bit of a shout out. The patient advocacy team is out there. They have a table. So they would love to meet the parents, meet the families. So if you get a chance, go to the table. I'll be out there as well to meet and greet as well. So today I'm going to be talking about PTC's work in their gene therapy program in AS. And I think it's a little bit apropos for me to be up here. In 2011, we actually did publish the paper for the gene therapy that we put into AAV9 into a mouse model for Angelman syndrome, and recovered some of the defects in that animal model. And I think it's also apropos that it's an ASF meeting that we're here talking about this, because the ASF actually funded a big part of that research. So you can see how this has progressed. 2011 is almost a decade later, and now we're talking about trying to get this first in human. So the research pathway for gene therapy, and the slide is a really basic simplification of an incredible amount of work that goes into this. It starts off with pharmacological studies. Usually this is in rodent models for whatever disorder you're looking at. This follows for pretty much all gene therapy programs. You look to see whether there's disease improvement, whether you're seeing kind of efficacy. You also can do some safety studies as well. Rodents are really great for this. Because they have a 2 to 2.5 year lifespan, so you can see how that therapeutic affects that animal through its entire life. After the pharmacological studies, this goes into toxicology studies. So these are usually in larger animals, non-human primates, or some kind of a bigger animal than the rodent. These are incredibly important, incredibly complicated, and incredibly expensive types of research that needs to be done. But the pharmacology and the toxicology, they really need to be bulletproof before you can go into that first in human study. So I want to try and get into some of the data. This is a snapshot of the data and where we are in the preclinical study. This is our PTC-AS, is what we're calling our new gene therapy. This says V5 on there because the V5 is a little tag that we put at the end of the protein so that we can kind of follow it and identify where it is and where it goes. And so the vector distribution is in this mouse model is kind of where the vector goes. So if we put in the vector, can we get it everywhere? This is a global deficiency in UBE3A throughout the CNS. We want to see if we can get the gene therapy to go everywhere in the CNS. And indeed, we see a nice distribution throughout the major brain regions that we look at. We also look at a transgene expression. So we put the gene in, can we get to the RNA? Does it look or have the same profile as the vector? And yes, the transgene does. And then we want to see what the expression is, what the distribution of that protein is. This is where that little V5 tag comes into play. So we have an antibody for that V5. And you can see on the far right, the top, you shouldn't see anything, that's just our vehicle. But as we increase that concentration of gene therapy, you start to see more and more of that expression. And at the end, all that red that you're seeing is expression of the UBE3A from that vector that we put in. Now, it's been known for some time that neurons in the animal models look different between our wild type animals, or our typical animals, and our Angelman syndrome animals. We see this in both the mouse and the rat models for AS. You don't need to be a neuroscientist to see that there's a difference between those two on the far left, right? So there's less branching in the AS models. If you look really closely at that branching, which is where the dendrites are, you see less of these little kind of spines on the dendrites. Those spines are important. Those spines are where potential synaptic connections can be made. So if you have less branching and you have less of these spines, you have less of a possibility of making those synaptic connections. And we've seen a nice dose response model that when we increase our gene therapy, you start to see a recovery, not only of that branching, but if you look closely at the branching, you can see a recovery of those little spines as well. This is also kind of a snapshot of our gene therapy program and our preclinical work. What I want you to kind of concentrate on is the black bars are the Angelman mice, the white bars are typical or wild type, and then the blue, light orange, and dark orange are kind of our increasing concentrations of gene therapy. So what we're hoping to see is that what you see with the Angelman mouse model, when we start giving it the gene therapy, it starts looking more and more like our wild type or our typical. The top one is the hind limb clasping. I'm sure you've seen this where they hold the mouse up by the tail. Angelman mice have a tendency to clasp their hind legs together. Wild type mice don't, they splay out. And you can see in a nice dose response that we see with increasing gene therapy, we can recover that phenotype. I think Allison actually showed the rotarod and different types of testing that you can do to look at motor coordination and motor learning. When we do the accelerating rotarod as well, you see two different things, not only motor coordination, how long they can stay on before they fall off, but how much they can learn from one day to the next. So anything that we do motor coordination wise, you have a learning curve. And from day 1 to day 2, we should see an increase in that effect. And indeed we do. With our gene therapy, we can see that we're getting closer and closer to what wild type mice look like. Overall motor function and just general sensory function too is something that we look at. This is where the nest building comes in. You saw a little bit of that earlier where they put in a little nestlet. Here it's a little square nestlet that wild type mice will immediately kind of tear apart and make a nice little nest. If you look at the very bottom, you can see what a normal nest looks like. It's nice and round. It's got a certain shape to it, and that's what you want to see. AS mice are really not very good at this. And so again, what we see was with our dosaging of our gene therapy, we can see that we can start having the AS mice make the nests look like wild type nests. And then finally, we looked at synaptic plasticity, and I've talked to a lot of you about synaptic plasticity. This is mostly done in the hippocampus. This is an electrophysiologic way that we can look at, see how neurons communicate with each other. We can do a number of different tests, but this is one where we give a high frequency stimulation and then look at plasticity, or how well you can increase the efficacy in neurons in the hippocampus. Both the mice that are on top and the rats that are on the bottom show this defect in synaptic plasticity. And we see a really nice dose response rescue with that in our AS mice and closer to a rescue with our AS rats. So we are recovering that as well. This probably is a lot to do with what I showed in the previous slide with that alteration in those neurons. Neurons that don't have a lot of synaptic connections have a defect in synaptic plasticity. So this is a nice way that our gene therapy can recover that as well. So I know that was a very quick snapshot of what we've done preclinically. Just to let you know where we are right now, we've completed totally the additional preclinical testing that we need to determine things like route of administration and dose selection. We're currently conducting toxicology studies. So in that slide that I showed earlier as well, now we're in those large animals, non-human primates, to really do what needs to be done for safety and efficacy for first in human trial, which is that FIH. The other thing that PTC has done is we've established a manufacturing process right in Hopewell in New Jersey. So PTC-AS will be the first gene therapy that we're actually developing and manufacturing those vectors right there in New Jersey for. If you YouTube this, you can kind of see what that's all about. But it's a pretty interesting facility that PTC has put a lot of money into so that we can manufacture our own gene therapy. And I think if nothing else, it really tells us our commitment, or tells you our commitment for going toward a lot of different therapies in gene therapy, including Angelman syndrome. And then we continue to work with ABOM, and I think you can't overstate the importance of biomarkers and outcome measures when we start talking about going into clinical trial. I know it was a really quick presentation, and but I do want you to know, so a lot of people thought that maybe PTC had taken a pause on their gene therapy approach. I can tell you that that's not the case. We continue to work as hard as we can to come up with the first-in-class and importantly, safe gene therapy that we can bring to the patient in the clinical trial as quickly as possible. And so I'm here to let you know that we're committed to that. We're committed to working with the foundations to see that that goes through. And I just want to let you know that that PTC, we're all in as well, and definitely here for the families. So with that, I want to thank you. I want to thank the Angelman Syndrome Foundation for their invitation to come out. It's really exciting to see so many people here and exciting to see all the work that's being done by all these companies. So thank you very much.
Thank you, PTC. All right, next up, I'll invite Becky Crean to join us on the stage to give us an update on the Ionis clinical trial called HALOS for Angelman syndrome.
Good afternoon. Thanks for having me. I echo the other speaker's sentiment that it's just really great to be back in person. It definitely gives a different vibe and energy to being together and providing these updates. So I'm glad to be able to be here. Thanks for inviting me. And just on behalf of myself, my Ionis colleagues, and our Biogen partners here, we're really excited to give you a brief update about our program and take you through that. I have my, let's see, how do we do this? Okay, so before we do that though, I want to, I'll do a couple things today before we get to the program update. I know that's the exciting part, but before we get to that, I do want to spend just a few minutes talking a little bit about some aspects of clinical trials that maybe we don't always talk about. So I'll talk about clinical trials in the context of rare disease and how sometimes they can take a little bit different approach than traditional clinical trials in drug development. We usually, as Allison had mentioned before, there's usually some changes in how we can combine the studies and try to get there faster and with fewer people involved. I'll also talk about how we can assess and determine if a drug is working in a clinical trial. I know that's always the big question is it working, and how do we know if it's working, and how do we know if that's meaningful to families and patients? So I'll talk a little bit about that too, and some ways that we can ensure we're testing it right, and also some things that can get in the way of us being able to test it correctly too. And then of course, I'll give an update on where we're at with our HALOS trial, which is, as you know, is a phase 1/2 trial testing Ionis's antisense oligonucleotide. So I know Allison talked about this a little bit earlier, but just to give you a backdrop of what traditional phases of clinical development look like, and I've been doing this for a while, and the steps haven't changed much. What I'm not going to talk about is all the preclinical work that gets you to the trial. We've had a lot of talks about that here and other meetings, but there is a very long and complicated process in identifying a drug, testing it in the animals, and kind of working its way through that phase. And then once we get to clinical development, that's really the development phase. And that too is a very long and complicated process. It seems simple when you hear phase 1, 2, 3. That sounds very simple, but it's quite complicated. So there are typically 3 main phases. Each phase has its own distinct question that it wants to ask. And so in the first trial, in the first phase, it really is a trial. The specific question is really about the safety of the compound. We want to start to get a sense of what that looks like in a human population. So these studies are typically very small. They're usually very short in duration. Usually only 1 dose is given. They're typically done in healthy volunteers. So before you can test them in your intended patient population with the disease, you first usually have to test them in healthy volunteers who don't have any diseases. And so then you can really get a sense of what the safety profile looks in that first phase. Typically, then you move on to phase 2. Those are usually a little bit longer trials and a little bit larger. You add a few more patients into those. And really you start to ask the question, you still look at safety, but then you start to ask the question about whether they're effective or not, if they do start to show some clinical benefit. Those trials can last anywhere from a couple of months up to a year. And then you get into the big trial, and that's the phase 3 trial. And this is the one you hear a lot about. You hear about, that's our goal for all of us, is to get to that phase 3 trial. And those are the larger trials that really provide the information that regulators need to decide whether they can approve this drug or not for use for everybody with the intended disorder. So that tests not only is the drug safe in a very large number of people, but also what is the clinical benefit to that. So those treatments studies are much longer and usually involve thousands of patients. Now, that's traditional drug development, and then there's rare disease development. And I think we've been hearing a lot more about the FDA sort of modifying their expectations for traditional drug development to really tailor towards the rare disease space, because they understand that the number of patients with the disorder is much smaller, so you can't do those large trials. And they also recognize that the clinical severity of the disease is such that we really do need to do this more efficiently. So typically what they allow us to do is, as Allison mentioned before, is to combine some of those first couple of phases. So you'll notice that with the trials that have been talked about so far today, they say they're a phase 1/2 trial. Ours is that way as well. And that really is that ability to combine phases and try to answer more questions in a shorter study, exposing fewer patients in those early trials. Usually what we can do is, if we do that combined trial, it usually sets us up, if we answer our questions correctly, we can typically go right into a phase 3 trial. And that looks like a traditional trial, but usually the numbers are smaller and perhaps the duration is a little bit shorter. So that's just sort of the difference between traditional drug development and then in specifically for rare disease. So we have as I mentioned, each phase is meant to answer sort of a specific question. And so the first question we always want to ask is this drug safe or not? Once we start to have a sense of whether it's safe or not, then we want to ask, well, does the drug work? Does it provide some clinical benefit? And so how do we do that? There's a number of different ways we can do that. And you'll hear a lot about endpoints or outcome measures. You heard before talk about the CGI. That's one particular way of measuring if a drug works or not. But those are used to evaluate if the drug is improving on how a patient feels or functions or survives. And they typically can be classified into two different types of endpoints. One is the objective measure or endpoint. That's a clinical endpoint which can be directly measured with the participant actively involved in the assessment. So things like, how far can a patient walk? How many seizures did the patient have? Things like that. Those have a lot of strengths to them. They directly assess the patient's abilities, the symptoms. They're not relying on subjective or opinions on whether somebody's doing better or not. One of the weaknesses to them, though, is they often don't tell us whether the changes are meaningful to patients and families or how impactful they are in the lives. I mean, being able, how far you can walk for 6 minutes doesn't necessarily translate in a meaningful way. So that's one of the weaknesses of them. The other type is a subjective measure. As you heard before, the CGI, that is a subjective measure. Now those involve personal interpretations or professional judgments about changes in a patient's lives. They often come in the form of questionnaires, either filled out by the clinician or study staff, sometimes by the parent or the patient themselves. One of the strengths is that it really does help us understand whether our treatment is meaningfully changing the life of the patient or the family. But again, the flip side is that the weakness is that it relies on a personal interpretation or a judgment call, and it's not a direct assessment. Ideally, in a clinical trial, you would be able to have endpoints that use both and that they both agree with each other. So we would like to be able to see changes on objective measures as well as subjective measures in a clinical trial. So that's why often if you're involved in a trial, you will get a number of different types of assessments. Some of them are objective, some of them are subjective, so that we can find, so that we can get agreement on both types. So those are ways that will tell us if our drug is improving a patient's lives or not. What that doesn't tell us is if a drug is better than not taking a drug, right? And so you hear a lot about placebo-controlled trials and how those will eventually be part of the clinical trials in Angelman syndrome. We haven't gotten there yet, but I think we will. These are placebo-controlled blinded clinical trials, and those really are considered the gold standard and the best way to determine whether a treatment works or not. So a placebo, when a placebo is used, that's where it's a treatment which does not have any active ingredients. So you often hear about a sugar pill, and whatever the type of drug it is, it gets disguised so that it looks, smells, tastes like the drug, but doesn't have any of the active ingredients in it. So they say that they really are the best way to determine whether a drug is working and if it's better than a placebo. Now, there is this thing about placebo effect. We've heard about it this morning. And I don't want to necessarily say that it's a bad thing. It's a really common thing in research. We all know about it. We kind of plan for it. But a placebo effect is when a patient has a response to a treatment, even when they aren't receiving the drug. And you think, well, how can that happen? Because they're not getting the active drug. But it's actually a real phenomenon. There's been a lot of research done on the placebo effect. People do have physiological and biological changes in the context of a study when they take a placebo. And so if you think about it, it makes sense. When you have a headache and you take a pill, an Advil, you expect it to make your headache go away. So if you take a pill that doesn't have the Advil in it, you're still going to have that expectation that it's going to take the headache away. And sometimes it actually does. It does make it more difficult to show the effect of an actual drug though. So we expect it to happen, but it does make the hurdle a little bit higher to show a true drug effect. So with a lot of these trials, and you'll hear sometimes, even if you have placebo-controlled trials, there usually is a point later on in the trial where that'll convert and everybody will get an active drug. It doesn't always happen, but don't be alarmed if you see placebo trials coming. Those typically will involve getting the active drug at some point, because as you've heard, it's really important in these trials to allow everybody the prospective benefit, and that's one way that we can do that. You'll also hear a lot about blinding in these trials, and that means that neither the clinician, the study team, or even the sponsor knows, or the patient knows what treatment arm they're in, whether they're getting the active drug or the placebo drug. That's one way that we have that can minimize the bias and reduce the placebo effect. And really see the true effects of the drug. It also allows the regulators, the FDA, to clearly evaluate the effect of a treatment. So these are some of the ways that researchers can control for influences in a trial that aren't necessarily due to the drug. So we can use the design of the study to help control for some of these factors. But there are some external factors that really can influence the success of a trial that are completely out of our control. And one of the ways that happens is through social media. I know we talk a lot about that. It's been around for a while. It's not new. We've known about this, but it has been gaining a lot more attention in the eyes of regulators and also researchers as well, that there actually can be some harmful effects on trials through the use of social media. I mean, on the one hand, it can have really, it can provide a benefit and a positive effects for families and patients who are in trials. It provides a community for them to share their experience or have a place where they can talk about their experience. But the flip side of that, I think the saying is every rose has a thorn. The flip side of that is that Too much sharing and too much discussion about a trial can also be harmful to the trial and can introduce a lot of bias into the trial and make it much more difficult for the study team to be able to assess whether their drug is working or not. So I won't go into a lot of detail about this. I know there's a whole panel tomorrow on the effects of social media and how it impacts these clinical trials. So I would encourage all of you to go to that. And I would just say that I know that some of these trials that are ongoing right now have specific criteria in their inclusion/exclusion criteria that prohibits people from using social media. And so if you see that in a trial that you may be interested in, this is one of the reasons that we ask that, is that it really does have some profound effects on our ability to really assess the drug. Okay, so enough about clinical trials. Now I can give you a brief update about the HALOS clinical study. So I don't know if you'll recall since the last time we talked about this, but ION 582 is Ionis's ASO, or antisense oligonucleotide, that's similarly designed to some of the other ASO treatments that you've heard about. And it's designed to unsilence the paternal allele, which would express the UBE3A protein in the neurons. The study is actively recruiting. It's ongoing. We launched it last December with the first site up and running was Roche. Roche is Rush. And this ASO, like the others, are administered intrathecally into the spine with a lumbar puncture. This trial is being done in individuals living with Angelman syndrome. With each intrathecal dose, you do get sedated in order to allow for the injection to occur safely. So in this trial, we're testing a broad age range of children and adults, age 2 to 50, but different age groups get staggered in at different points in the study, and I'll go over that in the next slide. We're enrolling about 44 subjects, maybe a few more than that. So it is a small trial. It is a phase 1/2 trial, as I mentioned. It is that combined approach, but it is still quite small. While you're in this trial, Angelman syndrome subjects will be given some tests in the clinic. Parents and caregivers will also fill out questionnaires at each clinic visit. And there's also some home assessments too that allow us to capture changes and things that are going on in your natural environment that might be more representative of how everybody's doing, that you don't always get to capture when you're in a clinic. So as I mentioned, it's a phase 1/2. The treatment length is about 3 months, and it includes 3 doses spread out over 3 different time periods. There is no placebo in this trial. It's all active drug, different dose levels, but the drug is given every 4 to 8 weeks over these 3 months. So again, the primary goal of this first study is to assess the safety and tolerability of the drug. But in addition, we're also going to measure all the clinical changes that we can capture in areas like communication, cognition, motor, sleep, seizures, and everyday functioning. We really want to take the opportunity to start to understand if the drug is having an impact on individual lives. So this is a global clinical trial across up to 13 sites. It's hard to read that map there, but it just shows that we have a number of sites in the United States, all of which I can say are part of the Angelman Syndrome Clinical Trial Network. And I know you'll hear more about that as well, but these are really sites that are what we consider centers of excellence and have the infrastructure and all the right people in place and all the right knowledge to really run these trials. They're an excellent resource. And so it's to put these first trials in the hands of these experts, we think is the best way to go. So we've been able to do that with the help of ASF and the PIs as well. We also have some sites that'll be in Europe. And all of these, we have many active sites in the US right now, and we hope to get the remainder of them globally open up by the end of the year. So in terms of what the study looks like and the timing of it, you can see the "We Are Here" mark. We've enrolled the first cohort A of patients, and they've been dosing for some time. We've started enrolling in the second cohort, which is cohort B. These are all in 4 to 17-year-old individuals with Angelman syndrome. And then you'll see later on, once we've gotten through the first 3, we'll expand the age range up and down. So we'll drop it down to 2-year-olds, and then we'll also go up to 50-year-olds later on. And you'll see a dark gray bar at the bottom. And what that's meant to represent is that we are starting to make plans right now to update the protocol to include that long-term extension. I think you just heard about that from Roche as well. We will be implementing that in this trial. And so once you've gone through those first 3 months of dosing, then you'll be able to what we call roll into the long-term extension and continue dosing for a much, much longer period of time. And that'll allow us to collect more long-term safety data and also more clinical data as well. So the study's progressing well as planned. As I mentioned, we have enrolled individuals so far and no safety signals have been observed. And we think adding that long-term extension will provide additional benefit to the families and also allow us to better assess the compound. So with that, I'd just like to thank everyone for your time and listening to the update. I mean, you see all the groups and collaborators on the bottom half of that screen, and we really couldn't do this without your participation and that of the foundations and organizations as well. We are here because of your commitment and your involvement and your participation in this process, and so we thank you for doing this along with us. That's it.
All right, thank you, Becky, and the entire Ionis team. Our last community update today will come from Neuren Pharmaceuticals, and I believe we'll be joined on Zoom by Nancy Jones.
Hello, everyone. Very pleased to be able to join you today, and thank you for the organizers for allowing us to participate virtually and be able to provide you this update. We're very pleased to be able to participate and be with you today. I'm here with my colleague actually online, Larry Glass, who's our Chief Science Officer. I'm also representing the other members of our team. And we are a small specialty pharmaceutical company, and we are particularly focused on developing treatments for rare neurodevelopmental disorders. And we all have a personal and professional interest in focusing on treatments for unmet medical needs. This is a passion for all of us. And our pipeline, as you can see, we've got 2 drugs in development, and they're all for rare neurodevelopmental disorders. And I just want to highlight that for our other compound, trofinetide, our Rett syndrome program is already completed a pivotal phase 3 and has been submitted by our US partner, Acadia, for a new drug application for an approval for FDA. So it's just with FDA and waiting for their review. And our program in NNZ-2591, which I'm going to talk about today, is in the development of phase 2. So the progress since we had our last update at the meeting last year is that we have gotten approval of our investigational drug application, which allows us to progress with our phase 2 in Angelman. And today I will give you, because there's some new people probably attending the meeting, an overview of our preclinical studies and our phase 1 study, as well as an update as what's going on with our phase 2 study. So Alice actually gave a little bit of a preview of NNZ-2591, and sorry, it doesn't have a quite snappy name yet, but it's good also to understand a little bit more about what it is, to be reminded that there are lots of different types of cells in the brain. So we talked, a lot of people have been talking a lot about the neurons, which you can see in this picture here in orange, and they're the main cells that communicate with each other. You've got the synapses, which is the connections, and those dendrites, which receive the messages. But there are also other supporting cells that help with the growth and maintenance of those. You can see that little brown one called microglia. And all of these cells, the neurons and the supporting cells, produce insulin-like growth factor, which is an important growth factor in the brain, which is responsible for the growth and maintenance of the cells. And when it acts in the natural state in your brain, it actually breaks up into smaller pieces to do its business. And those are called metabolites, these smaller pieces. One of those is called cyclic glycine proline, or CGP, and this metabolite is responsible for maintaining the sort of the balance between how much IGF-1 you have in your brain to make sure you have the optimal amount. And what NNZ-2591 is a synthetic analog of cyclic glycine proline. So what that means basically is that it has the same molecular structure as cyclic glycine proline, but it's got one small change to the molecular structure, and that makes it better to use as a medication. So that means it can still have the same function of what the naturally occurring metabolite does, the cyclic glycine proline, meaning regulating the amount of IGF-1. But it has all these properties you see on the right, that it's 100% bioavailable, which means all the drug is able to be used and absorbed. It crosses the blood-brain barrier. And then it also has good, what we call pharmacokinetic properties, meaning how the drug is used and absorbed. So that change makes it possible to be used better as a treatment. So particularly, what is it actually doing? So it's improving the problems that you see if you've got disorders of synaptic function, or the connections between the brain cells. So if you've got abnormal function of the microglia, that supporting cell that I showed you in brown, that maintain the connections between the synapses, you can get impaired signaling, you can get an increase in these inflammatory molecules, and then lower amounts or sort of lower availability of IGF-1. And what preclinical studies with NNZ-2591 have shown is that it can restore all of these dysfunctions. And it only affects the cells that have the impairment. It doesn't affect normal cells. This is state dependency, which is important with respect to safety. So it's not having any adverse effect on normal cells. And this is an example of the results from studies that's just in another model of another NDD, but it's just showing the effects that you can have. In this case, you can see these are with respect to dendrites. In this case, excessively long dendrites with treatment are sort of restored to normal. And here again, seeing those excessively long being restored to normal like the normal ones. And then with the IGF-1 levels we see here, this is in a wild type mouse that doesn't have any dysfunction. And the knockout mouse has lower amounts of IGF-1. When it's treated, those levels sort of go back up to the same level of the wild type. And in Angelman syndrome, we know that there is problems with synaptic plasticity in terms of the connections, the strengths of the connections between the neurons, and abnormal morphology and density of the dendritic spines in the preclinical models. So this is actually something that would be relevant for Angelman from the cellular level. But we also have done, I think also as Alice mentioned in her presentation, we have done a mouse model of Angelman syndrome looking at the sort of phenotype in the mouse model. And this is actually the results from that, which you saw sort of briefly in Allison's presentation. And what you're seeing is these are each of the tests that were done, sort of analogous behaviors to symptoms that you may see in Angelman. The wild type mouse, or the unaffected mouse, is always in sort of black or gray, and the blue is the Angelman mouse. And you can see on the left is pre-treatment. So in all the cases, you're seeing a difference in the types of behaviors that you're seeing and what's being tested. And when they're treated, you're seeing that it's rescued. So now the Angelman mouse is looking exactly like the wild type mouse. And another important feature, which I talked about, the state dependency, you're not seeing any kind of negative effect of what's happening to the wild type mouse. They're not being negatively affected by getting treated. So in sum, what did the preclinical result findings tell us? One, that the NNZ-2591 can have an effect on the brain cells. It can improve analogous impairments in the mouse model. And then what we saw in the mouse model is that there's a potential for treatment across a range of symptoms. So what have we done in terms of the human population in terms of safety? So we have done a phase 1 study looking at safety PK, and this was actually in healthy adults. And it was 2 phases that we looked at: a single dose and looking at the safety of that, and then looking at twice daily dosing with the NNZ-2591 or placebo over 7 days. And on overall finding is that it was a good safety profile. All the adverse events were mild or moderate, and they resolved during the trial. There were no severe adverse events, and there were no serious adverse events, and there were no findings on lab tests, vital signs, or cardiac tests. And the PK profile was predictable, which is what you want. Essentially, it's saying that the drug is behaving the way you want to see it in terms of how it's being used or taken up in the body. So our next step then is the phase 2 study for Angelman syndrome. And we have received, as I mentioned, the approval for the investigational new drug application for the US with the FDA. And the study is open for enrollment at the Children's Health Queensland Hospital, which is in Brisbane, Australia. And we're expecting the top line results in the first half of 2023. The phase 2 study is a small to moderate size study, and our aim is to evaluate safety and tolerability, but we're also exploring the potential effects of the drug. It is an open label study, which means that everyone is getting the treatment. The treatment period is 13 weeks. It's a liquid formulation, so it's oral dosing that's done daily. And there is a combination of in-clinic visits and in-home nursing visits to make it convenient for the family. And I mentioned the primary aim is the safety and also the pharmacokinetics to understand how the drug is absorbed and distributed. But we're also going to be looking at the clinical response to the drug. And you can see on the list of we're looking at a range of symptoms, and that is based on what we were seeing in the preclinical model, but also based on what we have learned from the work that you guys have done as a community in terms of what types of things are important to families and individuals with Angelman syndrome. And this is going to inform what we will do in the larger phase 3 studies to follow if the study is successful. These are some of the snapshot of the key inclusion/exclusion criteria. There's more detail on clinicaltrials.gov. It is sort of across the range of the full pediatric age range, 3 to 17, a diagnosis of AS with confirmed genetic etiology. Because it is a liquid formulation, being able to swallow the liquid, and seizures are allowed, but they just need to be on a stable profile and a stable treatment regimen. In addition to that, participants are going to need to do assessments at screening. There's going to be a review of medications and a medical history, which is also done to determine eligibility. So our study sites are all in Australia, and I do want to note that we are actually an Australian-based company. There are 3 sites. And as I mentioned, the Children's Health Queensland Hospital is open for enrollment. And as the other sites get open, their information will be updated on clinicaltrials.gov. We also have a study-specific website, and you can check that out. It has a lot more information about what is involved with participation, a little bit more about the drug and what the study is about. And if you want to be updated as things are, new sites are open, we do make periodic updates about the study, and you can sign up on neurenpharma.com to get email alerts if you want to be updated when we have new updates. So I wanted to thank you now for your for listening, but also more importantly, for all the work that you as the community and the organization has done. The design of this study is very much based upon direct input that we've gotten from members of the foundation, but also on the work that you guys have done in the community and natural history study and surveys that have been very important for us to understand what's important to families in designing this trial. So thank you very much.
Thank you, Nancy, and the entire Neuren team. I'm going to go ahead and invite all of our speakers up to the stage, and we'll have a quick panel discussion before we need to move on to some breakout sessions. So come on up. Don't be shy. I won't bite. All right, we're going to ask a few questions that we frequently get at the foundation, and so I'd like everybody to just take a seat, and then the way that we'll do this is I will direct a question to a specific individual, but I'd like you all to feel comfortable to weigh in on any question, okay? You might all have different perspectives, and all of those are valuable, all right? So first and foremost, thank you all for being here. Nancy, you're still with us here on Zoom, perfect. I guess I'll start with our Roche team. And ask you, and again, I'd love input from everyone, but what are some key factors that you feel like are really critical for ensuring success of these clinical trials?
Thank you. Can you hear me? No.
That does not seem to be on.
Can you hear me now?
Yes.
Cool. So yes, thank you for that question. So I think we have talked about a lot today about placebo response and things that go into that direction. And this is really key for us. So this is what we are concerned about. And in order to make these programs successful, ultimately, we need to all manage expectations. We need to manage expectation bias. And really that's what we try to promote. We do our piece to try not jump to conclusions prematurely, to protect the studies, protect the integrity of the studies, and as necessary. And only once we can do proper statistical analyses of these studies, only then we will really publish results. And from the community side of things, I think what can be done to make these programs successful, and we've all talked about various aspects of this, like Becky and also the other Becky talked about social media and how we should not further inflate expectations. Also from a family perspective, I just would like to re-emphasize that as a family, when you're on a trial and you see your child improve, you won't really truly be able to know what part of it is a drug effect and whether you are also seeing some kind of placebo effect there. So it would be just very beneficial overall to just, keep this for yourself for now and allow the group effect to occur and really, leave it to the statisticians and the data people behind the scenes to then finally assess what the true result really is. So that's what's important to us. So do you want to add?
You have a microphone. Great. Thank you.
Yeah.
Go ahead.
Well, I also want to add very quickly that it's really critical to engage the caregivers, the families, and those people that are really responsible and helping us cater to the patients that we are trying to deliver value to. They are the experts, and we need to engage them as early as possible, listen to them, learn from them, to really understand what the pathways are as we try to develop cure for this Angelman syndrome. I'll give a case in point. Within PTC, as we are seeking to really ramp up efforts for our clinical trial readiness and to chart a course forward, we, a couple of months ago, had to convene a caregiver focus group to listen to them, learn from them, and really understand the things that are most important and critical, for us to think about. We had a form of proposal about the initial thoughts we are having about our clinical program, but learning from them and hearing from them, because they're the ones that live day to day with these patients, was really very insightful. And we are really going to harness all of those lessons and the learnings we've received to know how to think about our program.
Absolutely. Thank you for that.
Yeah, I also, I echo what everyone is saying, and also want to underscore the importance of ensuring that we've learned from the earlier studies and adapt accordingly as we advance to the pivotal phase of the study. And to your point, learn from the families what is feasible and keep the study as simple as possible, because it's a great commitment for the participants as well as the caregivers and families to participate, and maintaining that commitment is important as well. And we do provide support in the way of travel and other means so that it's to minimize the the burden or the additional dimension that now is introduced to the family, and as well for the sites to make sure the sites have the appropriate support so they can conduct the study in the best possible way.
Thank you all for recognizing the burden on families for participating in clinical trials. Becky, all right, you guys have told us that we shouldn't turn to social media to learn what it's like to be in a clinical trial. So where can families learn what it's like to participate in a clinical trial?
Can you hear me?
Yes,
Okay, I'm not going to say social media.
yes, not social
Yeah.
media.
You know, deciding if participating in a clinical trial is right for you and your family is a really big decision. It's an important one, and it shouldn't be taken lightly. There's a lot to consider around it. And I think first and foremost, you have to go into it understanding that it's not treatment. It's an experimental study to understand if a drug works or not. And there's risks involved with it. And those risks change depending on what type of study you enroll in and where they are in their development. But I think I think understanding that that's the goal of a clinical trial is something that you have to consider in making this decision. We talk about treatments a lot, and sometimes we use that term interchangeably, and I'm at fault at that too, but I think we do need to take a moment to recognize that these are experimental clinical trials. So there's risks involved, and you have to be able to weigh those risks appropriately. And one way to do that is really through educating yourself. And there's a number of ways besides just social media on educating yourself about trials and what that means. I mean, again, each phase will ask a different question. And so your involvement will be different depending on what the phase of the trial is. I mean, your best resource is your local clinician. All physicians are aware of and understand how clinical trials work. Even if they're not involved in the clinical trial themselves, they understand the process and they can at least give you some basic information about what it might be like to be in a clinical trial. And then to get more specific about the specific ones in Angelman syndrome and the ones that are going on now, then you can reach out to the centers of excellence and all of those clinics that you highlight on your website. Your website is an incredible resource for families to talk about what it's like to be in a trial, what could be expected of you, what your obligations are. Those are the resources you want to tap into through your Angelman community, the foundations, the clinicians in the trial network. I think those are good places to get accurate and clear information about what it's like to be in a clinical trial. And then just understand that it really is an agreement. If you decide to go down that path, it is an agreement between you and the study team that you're both going to make this commitment together to go through the protocol as it's designed, for good or bad. We don't always understand why certain things are asked at certain time points, and usually we have a reason behind it. We go into it having a reason, but just understanding that you're making that commitment and you're going to take part in this collaboration and this relationship along the way to further advance the research. So
Great.
just be aware. Yeah.
Thank you. Nancy, I have a question for you. What can families do from your perspective while they're waiting to participate in a clinical trial?
Yeah, thanks. Yeah, it's a good question. And I think some of the things we've already touched on is an important point. We're generally speaking, informing yourself as much as possible about what's involved. And sort of there's two levels of it. If there, for example, there's a trial that you're interested in and it's already open and you're waiting for your site near you to be open, there may be quite a bit of information already on clinicaltrials.gov. There may be sites that you can contact directly. Again, even if you're just interested in a trial, if the site has a contact information on clinicaltrials.gov, you can contact them and find out, well, what's expected? What will it be actually like? And they can give you a more day-to-day sense of what is it like to get there? What is it what's going to involve? How much time is it going to take? And you can learn a little bit more about that to sort of prepare yourself. And also in terms of inclusion, exclusion criteria, sometimes studies may have specific criteria about stability of certain treatments, exclusion of certain treatments. So finding out about that ahead of time as maybe you're waiting for a site to open is possible if the site has, or the study has information already posted, for example. If it's something where maybe something is not yet in it's a type of trial that you're waiting for that's not yet in the clinic, you can sort of, as I think the whole panel's been discussing, learning more about the process of what it means to be in a trial, and finding out that general information. Your website actually is really great. It goes through all those different components from the time you're in a trial to the time of the planning of it. And I think that can go a long way so that the families feel ready when the information opens or the trial is ready to open, that they have an understanding of what they might need to be able to do to participate.
Great.
I wanted to add a couple things as well. As families are waiting, there are, as we've heard throughout the afternoon, a number of natural history studies and survey studies in which participants can engage. And that is a way also to be connected more broadly with the community and then learn more about the possibility of clinical trials going forward.
Great. It's a good plug for the natural history study and some of those other observational studies. All right, our last question, and this is something that's weighing heavily on the mind of the community, and I'll give you each an opportunity to chime in here, but what, in your opinion, or from your perspective, is being done to maximize access to clinical trials for the AS community, especially those who are not necessarily connected to an AS specialist or hospital, center of excellence?
I can start. I mean, access is probably going to be defined differently depending on what phase of development that the field is in and where things are at. I think we all recognize the numbers. I mean, there's way more Angelman syndrome families than there are spots in any clinical trial right now, even combined. And so access, so there's going to be limited access in these early days. I think it'll get larger and broaden access as we get further on in development. But I think that's an important piece to recognize. Access is also going to partly be determined by what the question is for the study and what the inclusion/exclusion criteria are. That'll naturally open or close access to certain families. I know for our trial, even though it's a small phase 1/2, we have a big global presence. And so one of the ways we're trying to ensure access, even in these early phases, is to try to have as big a global reach as we can, even though that means more limited access here in the United States, but it does open up access globally. And as Allison mentioned, we're all one big family, and so everyone should have access. So that's one way that we're trying to do that.
Go ahead.
Yeah, just adding a few points. That's basically also true. All of what you said is basically also true from my perspective or from the Roche perspective. But one thing that we also do is to really keep the communication channel open. The engagement with the advocacy groups and also with the families directly is really important to us. So our patient partnership director on the team and the team that's behind engages with families directly. So we also have a website that's called For Patients. So if you type that into Google, Roche For Patients, you'll be there. And you'll find clinical trials where they are conducted, and you find the Angelman clinical studies there, and really, really valuable information, a very rich source. Also, we can be found and contacted directly by families. So that's important. And also, like you said, Becky, we really are committed to make this a very global program. We really have plans to expand, both in terms of an exclusion criteria, but also in terms of where the trial is running, in terms of locations, very globally. So absolutely, we will try to reach everybody who wants to be on these studies.
Great, thank you. Yes, I agree with my colleagues as well, and want to add, as we have conducted and do conduct trials for patients with rare genetic diseases, often, to your point, the patients aren't right near a center where the study is open or a center of excellence where that's an opportunity. And what we have done, as I mentioned earlier, where there may or may not be access in a given country, we've provided the support, travel, et cetera, for patients to go to another country to be able to participate. And that is a tradition that we have that we will continue, and very much acknowledge and plan as well to do so going forward for the Angelman community.
Wonderful. Thanks, Camille.
In a similar light, as we are preparing to go into the clinic, we are thinking about a concept where we'll have something that's like a hub and a spoke paradigm, where we'll have connections to the epicenters of care, where you have the experts globally, and for patients from their local settings to have a way of having those connections, reaching those experts or be able to really deliver the care. And we'll make it a concierge kind of delivery of research, and hopefully we'll be able to get the cure delivered.
Wonderful. Well, on behalf of the Angelman Syndrome Foundation, we thank you all so much for your commitment to our communities and for being here. So thank you.
Thank you very much.